Welcome to The Explainer. Today we are diving deep into a core GCSE required practical, finding out which antiseptics are the most effective germ killers. Now this one is a really big deal for your exams, so let's jump right in.
So, let's think about this. How do we figure this out scientifically? I mean, if you've got a bunch of different cleaning products, how can you prove which one is actually the best at stopping germs? Well, that is the big question this entire practical is designed to answer. Okay, before we get into the step-by-step method, it's so important that we understand why we're even doing this. What's the fundamental scientific idea we're actually putting to the test here? First up, a really key distinction. This practical is all about testing substances that stop microbes from growing on surfaces. Think your kitchen counter or even your hands. That's what antiseptics do. They are totally different from antibiotics, which are the medicines you actually take to fight bacterial infections inside your body. So the goal here is to stop, or at the very least slow down, this incredible process. Bacteria multiply through something called binary fission. Basically, they just split in two. And under the right conditions, a single bacterium can become millions in just a matter of hours. It's kind of wild. Our job is to figure out which antiseptic is the best at throwing a wrench in that rapid growth. Now, when you're working with microorganisms, you have to be incredibly careful. And this isn't just about getting accurate results. It's a huge matter of safety. That's where something called aseptic technique comes in, and it is crucial. So why is this so critical? Well, it boils down to two big reasons. First, safety. We only want to grow the specific bacteria we're studying, not some random, potentially nasty microbe floating in from the air. And second, validity. If our experiment gets contaminated, our results are pretty much meaningless. We won't know if the antiseptic worked or if something else just swooped in and killed the bacteria. This process is absolutely key. You start with sterile equipment, a clean slate. You flame your inoculating route to kill any stray microbes before you transfer your bacteria. You tape the lid, but, and this is important, not all the way around. You need to let oxygen in, but keep airborne microbes out. And finally, you store it upside down. Why? To stop any condensation from dripping down onto your agar and ruining your beautiful bacterial lawn. And here's another huge safety point, straight from the AQA specification. We incubate at 25 degrees Celsius. So why not warmer, like human body temperature? Well, 37 degrees is the perfect temperature for growing pathogens that can be really harmful to us. So by keeping it cooler at 25 degrees, we encourage our bacteria to grow without accidentally culturing anything dangerous. Right, you've mastered the safety. Now for the main event. Here is the breakdown of how you actually do the investigation, from start to finish. Okay, you start with your agar plate, which is basically a jelly packed with nutrients for bacteria. You create an even lawn of your chosen bacteria across it. Then, you take little paper discs, soak each one in a different antiseptic, and pop them on the agar. And here's the crucial part. One disc gets soaked only in sterile water. This is your control. It proves that it's the antiseptic causing the effect, and not just the paper disc itself. After 2 days of incubation, you're going to come back to a very different-looking petri dish. But a cool-looking pattern isn't enough for science, right? We need to turn what we see into cold, hard data. This is the absolute key term you need to know, the zone of inhibition. It's that clear ring you see around some of the discs. You can almost think of it as a kill zone. It's where the antiseptic has seeped into the agar and completely stopped the bacteria from growing. And honestly, this is the core principle of our whole analysis. The result is simple and super visual. The bigger the clear circle, the more powerful the antiseptic. A tiny zone means it barely worked, and no zone at all, like you'd see around our water control, means it had zero effect. But we need to quantify this. In science, we don't just say bigger or smaller. We need numbers. So we measure the diameter of that clear zone, we have it to get the radius, and then we plug it into this classic formula to calculate the area. This gives us a really precise number to compare how effective each substance was. So a good results table will look a little something like this. You record the diameter, you calculate the radius, and then you work out the area for each antiseptic. You can see right away that antiseptic B, with an area of 314 square millimeters, is way more effective than antiseptic A. And the control, just as we'd expect, has an area of 0. Perfect.
Okay, so you've done the practical. You've got your results. Now, how do you turn that into top marks in an exam? This is where we connect the lab work directly to what those examiners are looking for.
Oh, this is an examiner's favorite question.
They want to know if you can actually think like a scientist. What factors did you have to keep exactly the same to make sure your test was fair?
Let's break it down. The one thing you change on purpose, that's your independent variable. In this case, it's the type of antiseptic. The thing you measure as a result, that's your dependent variable, the area of the zone of inhibition. And literally everything else has to be a control variable. You've got to use the same concentration of antiseptic, the same type of bacteria, and the same incubation time and temperature. If you change any of those, your comparison isn't valid anymore. Now, another key area for evaluation questions is error. Random error is when your results are a bit all over the place, maybe because your measurements were a bit inconsistent. You fix this by doing repeats and finding an average. Systematic error is sneakier. That's when all your results are wrong in the same way. Maybe your ruler is faulty or something. That means you have to fix your actual method or your equipment.
So, if an exam question asks how you could improve this investigation, here is your checklist for top marks. First, say you'd repeat the whole thing and calculate a mean. Mention that you'd look for and ignore any weird, anomalous results that don't fit the pattern. And finally, hammer home the point about keeping all those control variables constant.
Finally, let's connect what you did on a bench in a school lab to one of the single biggest challenges facing global medicine today. This isn't just an exam topic, folks. This is real-world science in action.
The exact same method that you use to test antiseptics is used by scientists all over the world to test new antibiotics against dangerous evolving superbugs like MRSA. You see, bacteria are constantly mutating, and some of them develop resistance to the drugs we rely on. This practical is a mini version of the vital work scientists do every day to try and stay one step ahead.
And that leaves us with a final thought. Your experiment gives you a snapshot of what works today, but what about tomorrow? As bacteria evolve and become resistant, the results of this very experiment could look completely different in just 10 years. It's a constant, critical race between scientific discovery and bacterial evolution.